On the right, you will see a DNA molecule with 3 gene sequences, genes 1, 2, and 3. A partial nucleotide sequence from gene 3 is at the top in dark blue, with the 3' to 5' sequence labeled as template strand. Transcription of the template strand produces the messenger RNA sequence in red, which is a 5' to 3' sequence of nucleotides. The next step is translation, whereby the nucleotides will be read codon by codon on a ribosome, and the amino acids that the specific codons represent will be joined together to make a polypeptide. Let us now go to the transcription process in more detail. We may divide transcription into the following stages. The first stage is initiation. The second stage is elongation, and the third stage is termination. The transcription process is directed by an enzyme named RNA polymerase II. The enzyme recognizes a sequence in the DNA to which it attaches and begins transcription. The sequence is called the promoter. In eukaryotic cells, there are several specialized proteins that help the RNA polymerase II locate the promoter area. These proteins are referred to as transcription factors. If you will examine figure 17.7, you will see the initiation being illustrated. The egg-shaped RNA polymerase has attached to the promoter sequence in the DNA. The sequence of DNA that is transcribed into an RNA molecule is called a transcription unit. In figure 17.7, the elongation process is being illustrated next. The RNA polymerase unwinds the DNA, breaks the hydrogen bonds, separating the two strands. The RNA transcript is synthesized in the 5' to 3' direction. Notice that only one strand of the DNA is transcribed. This is called the template strand. Figure 17.9 is the next illustration to look at. This is a larger enlarged version of the elongation process. Examine this more detailed picture of transcription. Notice that DNA template strand is 3' to 5' and the newly made RNA is 5' to 3' in direction. The RNA polymerase II is the large, rather egg-shaped object surrounding the separated DNA strands. Termination of transcription is the last stage. The mechanism of termination is different in bacteria, which are prokaryotic cells, and eukaryotes. In bacteria, the polymerase stops transcription at the end of the terminator, and the messenger RNA can be translated without further modification. In eukaryotes, RNA polymerase II transcribes the polyadenylation signal sequence. The RNA transcript is released 10 to 35 nucleotides past this polyadenylation sequence. Figure 17.7 illustrates the initiation, the elongation, and termination. With the termination at the bottom, the RNA transcript with termination is released, and the RNA polymerase II detaches from the DNA. In a eukaryotic cell, the primary transcript must be edited on a spliceosome before it can leave the nucleus and go to a ribosome for translation. Translation will be discussed in a later series.